So before that glitch just happened, we were discussing how when trying to determine that a compound is mutagenic, one might think you could take that compound and say, for example, mix it with some bacteria and then determine if the bacteria are mutagenized or changed into from one genotype into the other. The problem with that is that compounds that are not directly mutagenic can be processed in your body into mutagenic compounds by metabolism. So, for example, if you're talking about, and this isn't to say that it's mutagenic, but one could maybe wonder if it is and test it. If you thought that sodium benzoate in your soda was mutagenic, you might test it on bacteria. And you might find that it's not mutagenic, but it might be the case that the sodium benzoate is broken down in your body into compounds that are mutagenic, and that could be very harmful. So what you need to be able to do is you need to be able to figure out a way in which you can test a compound, not that it's simply directly mutagenic or whether it's actually indirectly mutagenic through the breakdown products that arise from its being metabolized in your body. And the way that that's done is using the Ames test. And the Ames test basically simulates human metabolism by basically approximating what enzymes do in your body to compounds, not by doing this on humans because you would be able to do this, but by taking the liver of mice. And if you take the liver of a mouse and you grind it up, you can basically sort of simulate the metabolic process in which all of those different enzymes are going to break down the compounds that are potentially of interest with respect to mutagenicity. So here we have a mouse, we take its liver out, we grind it up, we extract the liver enzymes, and then you can take your compound X, which is of interest to you, and you can combine it with this liver enzyme extract. Now, in addition to the liver enzyme extract, you also have a strain of bacteria, and what you're going to do is you're going to determine if the compound that you have mixed with these enzymes, which then breaks it down into many, many, many different other components, whether if that is added to a strain of bacteria that in this particular case is His minus, which means that it's unable to grow on media that lack histidine, whether or not the mutagenicity of this compound has the capacity to cause reversions. And so His minus getting converted into His plus would mean that mutations were causing a reversion to occur. And so, there's presumably some mutation inside the gene that is critical for histidine biosynthesis, and by mixing a mutagen with it, it reverts that mutation from the mutant version back to the wild type. And so here what you have is you recover on this plate 1, 2, 3, 4 revertants from the His minus state to the His plus state. Now you have different strains of bacteria. Okay, some of them might have mutations that are single point mutations, that if you mutate, say for example, the G back to the original T, then you can generate the ability to generate histidine, and you would then recover these colonies. That would be a point mutation, but there may be other strains of bacteria that have mutations in the histidine biosynthesis gene that instead of having single point mutations, but actually have maybe a one base pair insertion, which would be a frame shift. And so in this particular case, maybe strain 2 has insertions or deletions, then any reversion that one sees there might suggest that the compound, when mixed, of course, with these liver enzymes from the mouse, that this compound induces the ability for DNA polymerase to either skip or add base pairs. So, depending on the strain of bacteria, which has a particular type of lesion in His, either a point mutation or a frame shift, one can recover variants of this revertant strains that indicate that this compound might have a particular type of mutagenic capacity. Now, of course, you have to also have a control. So in this particular case, you have strain 1 or 2 mixed with the liver enzymes, and in this particular case, you don't see any colonies that revert from His minus back to His plus. So this suggests that this particular compound X, when mixed with these liver enzymes, has some mutagenic capacity. Here it produced 4 mutant colonies, and here it produced 1, 2, 3, 4, 5, 6 mutant colonies. Now, this compound X may not cause reversions in one of the assays with one strain, but may cause reversions in only the other strain. And so in this particular experiment with the compound aflatoxin B1, there are three strains, TA100, TA1538, and TA1535, and these strains might have different types of mutations in the His gene. And for example, TA100 might have a frameshift mutation in it. Now, as you add the aflatoxin B1 dose in nanograms with liver enzymes to these 3 different strains here, you can see with the blue and the green strain, there's no additional excess recovery of revertance. All right, so there's no induction of mutation in this particular case. But in this strain, which I said I think had the frameshift, yes, it had the frameshift, as you increase the dose of aflatoxin B1 mixed with these liver enzymes, as you do that, you increase the dose, you increase the number of revertant colonies, which suggests that aflatoxin B1 in this particular scenario might have some ability to antagonize DNA polymerase and increase the formation of frameshift mutations. Now, different types of mutations have different types of names. We've been spending a lot of time talking about what we would call loss of function mutations, in which one gene is completely disrupted. All right, we often talk about that in the case of genes that are recessive, that have recessive alleles, and the recessivity is due to haplosufficiency. So one single copy of a wild-type version is sufficient even if there's one copy that's completely knocked out. You only see the phenotype when you have the complete null as a homozygote. That's a full loss of function, but one can also have something called a hypomorph, hypo meaning lower than, and a hypomorph means it has reduced function, so the gene allele doesn't function quite as well, but it's not a complete knockout. One can also have a hypermorph, which means enhanced function. An example of a gene that might have enhanced function is you can imagine that there's a protein that promotes cell division, and if this protein has been mutated in a particular way to cause a hypomorph allele, it might produce an excess amount of cell division, which would cause maybe cancer. And then finally, there's something called a neomorph, which is a gain of function, which means a gene might be expressed in some entirely new tissue. Now, one question that was thought about a lot in the early part of the last century was whether mutation arises, quote, when it's needed. Now, if one does an experiment in which you take, say, for example, 10 million colonies of E. coli that are sensitive to the T1 phage, meaning that when you expose them to the T1 phage, they die. If you took millions of colonies and exposed them to T1 phage, you will recover some phage that are resistant. So in this particular case, you have millions of colonies that are resistant to T1, that are sensitive to T1 that get killed by T1, and you'll recover some that aren't killed by T1, and so they would be called T1 phage resistant, or TON-R. Now, what is going on? So at the time, it was thought that one possibility is that the E. coli is responding directly to the phage. So the phage is inducing some sort of stress response. The bacteria, sort of freak out, not cognitively, but there's some mechanism in place that the bacteria induce mutations in themselves to become resistant to this phage. So it's an interesting idea, and it had to be tested because that sort of implies that mutation arises in sort of a directed way when it is needed. The way that this was determined as to whether it was true, it was shown to not be true. In fact, mutation is not directed by the organism's need. All right, and this was found to be true by Esther and Joshua Lederberg. Esther Lederberg developed a method called replica plating, and this is a critical part, is that if you have bacteria, 1, 2, 3, 4, 5 colonies, if you take a velvet stamp, which has been sterilized, one can copy this configuration of bacteria onto additional stamps. So you can stamp 1, and then 2, and then 3, and then 4, and then 5 different plates. But each of those plates will have the same configuration of the genotypes of bacteria at that particular particular formation. So the way that replica plating was used to ask this question about whether mutation was induced under stress or some other mechanism was to basically do the experiment with the sensitive E. coli, and the E. coli being sensitive were then, before the phage was administered to the E. coli, this plate here was replica plated here, here, and here. Now, if this plate was replica plated here, here, and here, and the individual colonies of bacteria might respond to the phage that is imposed upon them, under some circumstances, one strain might mutate. Under some circumstances, another strain might mutate, or another colony might mutate. And so what you would expect there, if the colonies are responding individually to the exposure to the phage, we would expect to see different patterns on each of these plates. So you might expect plate 1, all of these E. coli are exposed, and then this bacteria, and this bacteria might induce mutation, and you would see a pattern like such. And then on another plate, another set of bacteria would respond to the phage and induce mutations in them. And what you would predict under that idea, that mutation is directed by stress to, quote, help the organism, you would expect to see random configurations of bacteria on these plates. But that's not what was seen. What was seen is that each of these plates had the same exact configuration of resistant bacteria. So what's going on? What this tells us is that in the original plate, the resistance was already there before the phage were even put on the bacteria. So here you have this colony, this colony, this colony, this colony, and they are already resistant. And so by replica plating this, you put these colonies in this configuration, this configuration, this configuration, you expose them to phage, and all of them in that pattern survive. So what this tells us is that the mutation is not in response to the stress of the phage, but the mutation was pre-existing, in fact, and only revealed by the exposure to the phage. And this is a critical point in evolutionary theory, because what it tells us is that natural selection doesn't act to directly change an organism by causing an organism to respond to the environment. It's just that variants in the population are filtered out by natural selection, and it's the variation is pre-existing before the in fact stress is there, and the stress doesn't directly induce the mutation.